गुड मॉर्निंग एवरीवन। सो आज का हमारा टॉपिक है हिस्ट्री ऑफ डीएनए। डिस्कवर्ड कब हुआ था डीएनए और किसने किया था? सो फर्स्ट साइंटिस्ट का नाम आएगा, नाम है फ्रेडरिक मिशर, जिन्होंने एक स्टडी कंडक्ट करा था 1869 में। किसके ऊपर? फर्स्ट सेल के ऊपर। फर्स्ट सेल के ऊपर। अब ये फर्स्ट सेल क्या होता है? फर्स्ट सेल मीन्स कि इट इज अ कॉम्बिनेशन ऑफ डेड प्लस WBC. First cell is the combination of dead cell and WBC, white blood cell. So he was studying on nuclei of WBC. He found that a new substance in nucleus. He found a new substance in nucleus and he named it nucleus. And the function of it, the function of the new substance called nuclein is unknown during that time, and the structures also unknown. So he just named it nuclein. The second scientist was Frederick Griffith, who did a experiment in 1928. He was experimenting on Streptococcus pneumonia bacteria. He was experimenting on Streptococcus bacteria. Streptococcus bacteria, Streptococcus pneumonia bacteria that causes pneumonia disease. Yes. So he took two types of bacteria. One is harmless bacteria and the second one is deadly bacteria. When he mixed both of the bacteria, he found the all bacteria are deadly bacteria. So he concluded that there would be something happen in between the two bacteria. There would be something happen in deadly bacteria and harmless bacteria that converted all the bacteria into a deadly bacteria. So he just named the transformation principle. The transformation principle said something happen in between these two bacteria that convert the harmless bacteria into a deadly bacteria. Yes, so he just proposed a principle called transformation principle. Yes, and the third scientist was Oswald, a very Colin MacLeod and The last scientist was EMC Carty. He was also, they were also experimenting on some bacteria in 1944. He took, they took, they took two kind of bacteria. One is purified shell component of a bacteria, and the second one is tested tested cell component. Tested cell component means the bacteria which are just transformed. Yes, so they were crushed or mixed together and found all the bacteria are converted into tested bacteria. Means all the bacteria are transformed. So he concluded that there would be something happen in between them. There would be something happen in between the tested cell components and the purified cell component. So he concluded that the only things happen in the nucleus are only one component in the nucleus that can have ability to transfer the genetic information from a tested component to a purified component. So he just named that DNA has ability to transfer the genetic information from a tested cell component to a purified cell component. So all the bacteria components were tested component. Yes, इट मीन्स कि कुछ ऐसा हुआ होगा इन दोनों के बीच में, टेस्टेड कंपोनेंट और ये प्यूरिफाइड कंपोनेंट के बीच में, कि जितनी भी कंपोनेंट्स आई सेल की वो सब टेस्टेड की तरह बिहेव कर रही थी. ये मीन्स जो ट्रांसफॉर्म सेल्स हैं उनकी तरह बिहेव कर रही थी. तो उन्होंने क्या कैलकुलेट किया कि सेल में ही एक ऐसा चीज है जिसका नाम दिया डीएनए, जो क्या करता है, वो ट्रांसपोर्टर की तरह काम करता है, या फिर लाइक कार की तरह काम करता है, जो क्या कैरी करता है, एक जेनेटिक इंफॉर्मेशन कैरी करता है एक पेरेंट से दूसरे पेरेंट में, या फिर यहाँ ये हम बोल सकते हैं कि टेस्टेड कंपोनेंट से जो प्यूरिफाइड कंपोनेंट में जो जेनेटिक इंफॉर्मेशन ट्रांसफर हुआ वो किसकी वजह से हुआ, डीएनए की वजह से हुआ. तो क्या नाम यहाँ से इजाद हुआ, क्या डिस्कवरी हुआ, डीएनए का, कि डीएनए ही एक ऐसा यूकैरियोटिक सेल में या फिर एनिमल सेल में डीएनए ही एक ऐसा कंपोनेंट है जो क्या करता है, जेनेटिक इंफॉर्मेशन को कैरी करता है फ्रॉम पेरेंट टू देयर ऑफस्प्रिंग. सो ही कंक्लूडेड डीएनए is only things that have ability to transfer the genetic information from a tested cell component to a purified cell components. So after that, the fourth scientist was Erwin Chargaff. Erwin Chargaff in 1951 to 1950. In 1950, he told that DNA contain base pairs or bases or nitrogenous bases. Erwin Chargaff said that DNA contain bases or nitrogen bases that are of four bases, and they are adenine, guanine, thiamine, and the last one is cytosine. These scientists also proposed that these four nitrogenous base pairs bond with each other by a hydrogen bond in between them. Four types of bases that are adenine, guanine, thymine, and cytosine. Yes, and this, she took a photo of DNA. That means X-ray diffraction photo of DNA by using a famous photo called Photo 51. So she said that the DNA is a spiral in shape or spherical in shape. That means helical structure. Yes. After that, the sixth scientist was James Watson and Franklin Crick. In 1953, he uses the evidences of Franklin, the like X-ray diffraction photo of DNA, and published that the DNA is a double helical structure model. DNA is a double helical structure and published it. Later on, that publication was called as double helical model of DNA. Yes, so that is the history of DNA, who contributes the a lot of things in discovery of DNA. So thank you, class. We will talk later on the structure of DNA and DNA replication. So keep watching my videos. Do like, subscribe, and comment on my videos.